Hello everyone, my name is Jingjing Wang. The topic of this video is a blueprint of the human gut microbial genomic variations. I will present this topic from three parts. In the first part, I will give you a brief guide to metagenome-wide association analysis. In part 2, I will talk about current changes in metagenome-wide association analysis. In part 3, I will give you our solutions to these changes and some of the results. Now we come to the first part. Let's see what is MWAS. The full name of MWAS is Metagenome-Wide Association Studies. The main aim of MWAS is to use the metagenome data to identify associations between the human microbiome and several known covariates, such as some physiology data, the blood measures, diseases, drugs, lifestyles, and so on. To do a metagenome-wide association study, the key point is to know the microbial composition, which means to identify microbes involved in the metagenomic sequencing and quantify their abundance. There are 3 steps if we want to know the microbial composition. The first step is alignment, which aligns reads from metagenomic sequencing data to the species margin set. We can see in this figure, these are metagenomic short reads. These are species margins. A line means a species marked gene, and lines with different colors indicate marked genes of different species. After alignment, we can calculate the gene abundance based on the vertical and horizontal coverage. Then the taxonomic profiles are determined based on the gene abundance. Finally, we can get this taxonomic abundance table, which shows the microbial composition of metagenomic samples. We call this process as the metagenome profiling with species abundance. Now we come to the part 2, current changes in MWAS. Currently, the methods of metagenome profiling with abundance are commonly used in many MWAS papers and help to identify a lot of significant associations between human phenotypes and microbiome. However, there is a problem of metagenome profiling with abundance because it cannot establish a complete association analysis at the genome level. You can see in this figure, when we calculate the taxonomic abundance, we just align the metagenomic reads to the species marked gene, not the whole genome of the species. It will lose some information from the whole genome, and this information can be some important functional elements in genomes. Another problem is that it ignores the strain-level differences in the human gut microbiome. In biology, a strain is a genetic variant or subtype within species, and different strains of the same species may have totally different functions. For example, the species of E. coli has harmless commensal strains and pathogenic strains with ability to cause diseases in human hosts. So when we provide a metagenome, we also need to consider the differences at the strain level. You can see in this figure, a circle means a species. In the left, the number of these species is 1, but in the right, the number of these species is 3. This is the microbial differences at the species abundance level. However, in this figure, a circle, the species abundance in the right is the same as the species in the left, but in the right, the species has some changes and generates 3 different strains. That is to say, they are the same species but different strains. This is a microbial difference at the genetic variation level. So in MWAS, if we only consider the species abundance for association analysis, we'll miss information from microbial genetic variations. Now we come to our solutions and results. Here are 3 sections to illustrate our solution and results. First, we construct a human gut reference genome database. Second, we construct a human gut microbial genomic variations catalog based on the reference genome database. Finally, we compared the microbial genetic variation differences between the Chinese population and the non-Chinese population based on the human gut microbial genetic variation catalog. Now let's come to the first section, human gut reference genome database construction. This is a workflow to construct the reference genome database. On the one hand, we collected a lot of metagenomic samples and then used these samples to generate a lot of MAGs with the assembly and concatenation pipeline. On the other hand, we downloaded a lot of available isolate genomes and MAGs from public resources. Finally, we integrate all of these genomes together as our reference genome database. By integrating all of these genomes together, there are more than 270,000 genomes in our database, and these genomes can be grouped into more than 5,000 species clusters. Our reference genome database consists of 45 archaea species and more than 5,000 bacteria species. This is the phylogenetic tree of the bacteria species. Compared with other human gut genome catalogs such as UHGG, our database has much more high quality genomes. The quality score and N50 of representative genomes in our database are significantly higher than that of UGG. All of these findings indicate that we have constructed a high-quality human gut reference genome database. Now let's come to the second section, the human gut microbial genomic variation catalog. In this research, we consider 2 types of genomic variations. The first is ISMB, the single nucleotide variation. It occurs when a single nucleotide in the genome sequence is altered. The second type of genomic variation is structural variation. Structural variations are highly variable genomic segments, usually more than 1,000 BP within bacterial species, including deletions and duplications, which we call them deletion structural variations and the variable structural variations. In this project, we totally identified more than 50 million biallele ISMBs from more than 900 bacterial species across 14 phyla, and we found the top 20 species with the highest number of ISMBs were all species from the phyla of Proteobacterium, Bacteroidetes, and Firmicutes. We also identified a lot of deletion structure variations and variable structure variations from almost 2,000 species. And we found that the fraction of the genome region where deletion structure variations occurred was significantly higher than that of the variable structure variations. Now let's come to the last section, the microbial genomic variation differences between the Chinese population and the non-Chinese population. By alpha diversity analysis, we identified 140 and 54, 42, and 25 species whose ISMB, deletion SV, and variable SV diversities was significantly different between the Chinese and non-Chinese population respectively. All significant species in the Bacterotidae family had higher deletion structural variations based and ISMB based alpha diversity in the Chinese population. By the per-ANOVA analysis, we also found that the geographical location could significantly explain much more variance of the inter-individual microbial genomic variations across species than the age, sex, and BMI, which emphasizes the important influence of factors behind the geographical location, such as the diet, environment, genetics, and lifestyles on the microbial variations. Finally, by the genomic level association analysis, we also identified a lot of ISMVs and SVs that were significantly different between the Chinese and non-Chinese population. We also found that the associated SVs were enriched with the functions of drug resistance. For example, this is a VSV in the species of Blautia. It has significantly higher copy numbers in the Chinese population in both our discovery cohort and replication cohort. And this VSV was related to the drug resistance protein. Okay, that's all my presentation. Please feel free to contact me by email if you have any question. Thank you.